Yeah.
Yes.
Great. Thank you.
Yes.
Thank you. So let's start it. From the topic one, topic two, we talk about first the evolution of the circulatory system, and then the major components of a human circulatory system. And then in topic two, we mainly talk about the function, how to evaluate the function of the heart, which is the cardiac output. And the cardiac output have two major contributor. One is the heart rate, and another is the stroke volume. And so both the heart rate and the stroke volume, eventually they are achieved by two type of cells in the heart: cardiomyocyte, which is the mechanical structure that finally make heart contract, and then the pacemaker cells that give the electrical signal. And today we'll talk about both cells. And then let's first start with the cardiomyocyte. So cardiomyocyte, we already talked about in the lab. Cardio is the heart, myo muscle, cyte the cell, the heart muscle cell. Um, so before we even talk about the cardiomyocyte, so let's first go over the anatomy structure of the heart in a microscopic level. And again, we also talked about this in the lab yesterday. And this is showing a heart here, and this is a cross section of the heart. This red part, the thickest component of the heart, is made by the muscle layer, and this layer cell called myocardium. And the myocardium primarily contain cardiomyocytes, but not only that, it also contains the blood vessels and the fibroblasts that produce the collagen. And then on top of the myocardium or cardiomyocytes, these are two layers of membrane that we discussed yesterday. This inner layer, we call them the visceral pericardium. And it actually has another name called pericardium. The two names for the same structure. And then the outer layer, we call it parietal pericardium. And in between these two layers, there's a space that is filled with fluid, lubricated fluid, called pericardial fluid. And this structure is making this whole organ, this whole heart, when it beat, you don't really feel the friction. There's all about a fine lubrication here. And then let's go and focus on the cardiomyocytes. And then when we talk about the cardiomyocytes, and we would like to compare the cardiomyocytes with the skeletal muscles. So both cardiomyocytes and the heart muscles and the skeletal muscles, they are muscles. And in the lab, we also talk about the third muscle, the smooth muscle. Actually, smooth muscle is not the third muscle. Smooth muscle is the first muscle that's showing up in the evolution. And in the case of smooth muscle, this contraction molecules that we're going to talk about later existing there, but they are not organized. They're not aligned together. So in the smooth muscle, you do not see this layer by layer of the highly in order aligned molecules. And so smooth muscle, normally they are not very strong when they contract. And then the common feature between the skeletal muscle and the cardiac muscle is there is the structure called sarcomere. I think I saw something here. So the sarcomere, what is a sarcomere? Basically, do I have another thing? Yes. Basically, a sarcomere is, we'll talk about in the next slides, but sarcomere in this case is like a soldiers standing in a very, very aligned line there. And then, even though each single molecule is so small to recognize even under a microscope, but when they join together one by one, aligned very straight, you can see a pattern under a microscope, and this pattern called striation. And so both skeletal muscle and the heart muscle has this striation, has a sarcomere, so showing the striation. But so that's the common thing between the skeletal muscle and the heart muscle. And what are difference? What are the difference between the skeletal muscle and the heart muscle? That's how the muscles are organized. And just imagine any muscle, most any muscle you can think of. We can say a skeletal muscle is largely a linear. Skeletal muscle normally has only one direction, a contract, from longer to shorter. So it's a linear contraction. So in the case of skeletal muscle, all the skeletal muscle cells has another name called myocyte. So it's a myocyte, it's a muscle cell. And all the a single myocyte is actually a fusion of 100, even thousands of cells together. And they all fuse up into one fiber. And this each one skeletal muscle fiber is as long as a skeleton. And in these cases, when you contract, and then multiple fibers bundle together. So we contract all the fibers, contract, not every time all the fibers, but some of the fibers at least contract together. This is skeletal muscle. Which has a linear direction, and is a fusion of hundreds of thousands of muscle cells. And then you think about the heart muscle. The function of the heart is contract, is squeeze, becomes smaller. And instead of linear, the heart muscle more like forming a network. In this case, the fusion into a single fiber does not work efficiently for a heart muscle to contract, to squeeze the blood out of the heart. So in the case of the heart, the cells, the cardiomyocytes, do not fuse together. Instead, they each one of those averagely, I have the number here. I'm not sure my memory is correct. I think of averagely one single cardiomyocyte connected to, oh, I have here nine other cardiomyocytes. And how do they connect it? Just imagine, I guess you know octopus, right? So just imagine a group of octopus all joined together, hand by hand. Each one octopus has one tentacles, grab another octopus, and then it has eight octopus, how to call it, arms, 8 arms. And then so that each single octopus can grab another 8 octopus. And then each another one grab another 8 other octopus. In together, they join a network. And when all of those retreat their arms, this whole network becomes smaller. And you can imagine each single cardiomyocyte is like an octopus. The number is also similar, averagely 9 branches. So instead of arms, a cardiomyocyte has a branch, has not a branch, has branches. Branches reach out, and each single branch connect to another cardiomyocyte. In total, in all the cardiomyocytes basically connect together, likely forming a network. And actually, in the next class, we will further talk about it. It looks like a network, but actually, it's not really a network. It's more like a tree. More like a tree has branches, but this tree folding back to cover the entire heart. And then here we mainly focus on how does the two cardiomyocytes connect to each other. If you imagine an octopus, or how two human, how we can join up hand by hand, we hold our hand, right? But this is not the case in the cardiomyocytes. Cardiomyocyte does not really have a hand there. And what kind of a mechanism that can join two cardiomyocytes together? This is a structure that we talk about in the lab called intercalated disc here. Intercalated disc. And what happened here in the intercalated disc? In this case, the intercalated disc is something like a yeah, I cannot find an example, but they are just two surface, but they are not smooth. They have I don't know how to explain in English. So something up, something down, something like this, like two fingers, but it fit up perfectly. And then between these two cell memories, these are now you have the cell memory of two cardiomyocytes, and they are fixed together. And there are major three type of a cell-to-cell junction there. And one is called the gap junction. Here's the gap junction. Let me see. I have here the gap junction. Actually, I mentioned it first because we're not going further to the gap junction. Gap junction will be the focus of the next topic. And in this topic, we mainly focus on another two junctions. That's desmosome and adherence junction. Where are they? Somewhere here. Here, adherence junction and desmosomes. So what's the difference between the desmosome and the adherence junction? The desmosome is more like an anchor, and so our heart keep beating very very strong, but rarely. In the last week we in the lab, not last week, Monday and on the lab, we talk about some diseases in the heart, like a heart attack, like a heart fibrillation, and even heart failure when the muscle start to beat weaker and weaker. But literally, there's no kind of these diseases that the heart muscle to muscle junction is not strong enough. When the heart beat, and suddenly just disassemble by itself. So that's because the very strong connection between two cardiomyocytes in the place of the intercalated disc. And this strong connection, the anchor, strongly anchor the two cardiomyocytes together is the desmosome. Is desmosome. And then desmosome itself is only a passive anchor to make sure two cardiomyocytes always join together. And another thing is the cardiomyocyte is a motive, is a motion unit, a contract, and one cardiomyocyte generates some power, and this power need to be passed to the next cardiomyocyte. In these cases, how the mechanical force can be transmitted from one cardiomyocyte to the next cardiomyocyte through another junction. This is called adherence junction. Adherence junction is directly connection of the sarcomere from one cell to another, to the sarcomere of the next cell. Adherent junction. Now let's talk about what is a sarcomere. And I believe this one has been introduced last semester when you learned the skeletal muscle. And also, I believe at least some of you do not remember what it is. Now let's repeat. Let's review this knowledge. What is a sarcomere? And before we introduce what is a sarcomere, we have to introduce two types of big molecules, big proteins. One is called the fibrin. Not fibrin. Sorry. The actin, fibrous actin. One is the actin, another is called the myosin. So, do I have it? Somewhere in my slides. Somewhere here. I have a lot of work here. Myosin. So what's the difference between the actin and the fibrin and the myosin? So in these two cases, So in this case, the actin is the passive partner. The actin by itself does not really provide a power that drive the muscle contraction. And the power side is located on the protein called myosin. What happened here is that a myosin will drag will drag the actin and then push the actin inward. In this case, this is the myosin. This is the myosin, and this is the actin. So this is the resting status of the a sarcomere or muscle cell. In this cases, the actin and the myosin does not really have a large overlap. But when this muscle is activated, activated, the myosin will push the actin inward, and now they have a larger overlap between these two molecules. And then in the center of the sarcomere, there is a large bundle of the myosin. This is a bundle of myosin, another bundle of myosin, another bundle of myosin. And this large bundle of myosin has two heads, point to two directions of the sarcomere. And then in both directions, there's another group of actin. One group of actin or another group of actin. Here I'm showing the cross section of how the structure it look like. So in this case, if one this myosin is activated, then this myosin will drag the two group of actin proteins closer to each other. And then when these two actin group become closer to each other, there's not only that. So the actin fibers also connected to another layer of protein. This is a protein structure called Z disc. And the Z disc is actually connected to another memory of the cells. In these cases, so basically the myosin dragging in the center bring two actin groups closer. They bring the two Z discs closer. And when the two Z discs closer, the cell becomes shorter. That's the fundamental way of how skeletal muscle can contract and how the heart muscle can contract. And again, in this way, our action all is contraction. We actually do not have the capability of being longer, elongation out. We can only contract to pull closer. So next question is, how can this be activated? How can the myosin can be activated? One obvious thing is either in the heart or in the skeletal muscle. The myosin does not really always being active. It's waiting for the signal to be activated. And then how? So every semester I like to use this as an example. And have you heard about this story about Romeo and Juliet? Anyone have heard it?
Yes.
Yes. How they met each other? Do you know? Do you know how they met each other?
No. I see some movies, so I don't know.
Yes. So I don't remember in the original Shakespeare's book how they met each other, but in the movie they met each other in a party, in a big party, a party with a mask. And then I think that suddenly Julia lost his mask, and then Romeo saw Julia's face, and then they just fall in love, and followed by the tragedies. So, which means at the very beginning, if Julia does not really lose her mask, nothing that bad will happen. So, why I mention this one? Just imagine Mousen is Julia, and acting is I'm sorry, Mousen is Romeo, and acting is Julia. So they will fall in love or have the reaction once they see each other. You do not need to do anything. If you want the reaction, all you need is to remove the mask. So this is really what happened between the actin and the myosin. Myosin has the natural affinity or natural responding to the actin. So whenever the actin, the myosin really touch or see, they don't really see, but get close to the actin, real actin, they will automatically drag actin closer. However, so in the cases, but in when the muscle is in the resting status, the myosin do not see the actin at all because actin, we say like the Julia's face is masked by another protein called tropomyosin. Tropomyosin is another long protein, and its only function is lay on the surface of the actin and it blocked the myosin binding site on the surface of the actin. So in these cases, as long as you have the tropomyosin in position, myosin does not or cannot grab actin and push inward. So, and then another word to say so, as long as you remove the mask, remove the tropomyosin, and then when this myosin binding site on the actin protein is exposed, then you have this automatically contraction of the muscles. You have the myosin binding to the actin and drag the sarcomere shorter. So now, next question: how this tropomyosin can be removed from the actin? Tropomyosin is anchored to the actin by another protein. So tropomyosin does not attach to the actin by itself. It need another protein, like an anchor or tied to stay in the right position. This protein called troponin. So troponin can change its position. It's like a switch. You can put tropomyosin in the blocking status, or you can remove the tropomyosin in another status. So in these cases, if whenever you want the muscle to contract, you all you need to do is to activate the troponin to remove the tropomyosin from the actin, and then everything happens automatically. Nothing. So again, the only thing you need to do is to activate the tropomyosin to remove activate the troponin to remove the tropomyosin. So how you can activate the troponin, that's the next topics. And before we move into the next topic, we have there's one of the two diagnosis, clinical diagnosis stuff that I want to introduce in this class. Troponin. So in yesterday in the lab, and I think I mentioned this, maybe you still remember. So say sometimes people may have panic attack or just a regular chest pain. So whenever we have whenever we have any places in the heart that have reduced blood supply, we will develop a chest pain, very painful. However, not every time a chest pain leads to a heart attack. So what's the difference between a chest pain and a heart attack? And heart attack, more scientifically, they call it infarction. So how you define, how you determine between the chest pain and the infarction or heart attack? You cannot based on how painful it is. Sometimes some people feel more pain. So the key difference between a regular chest pain caused by reduced blood supply and a real heart attack is the cell death. So that's the fundamental difference between a regular chest pain and the heart attack. When the heart muscle, cardiomyocytes, start to die, we call it a heart attack. But clinically, how you can diagnose is really a heart attack. So when a heart cell died, this cell basically ruptured, have so many holes on the surface, and then these contents are released into the blood. However, in this case, all of those proteins, myosin, actin, tropomyosin, all of those are big proteins. Even though the cardiomyocyte died, these proteins will not be released out into the blood. The only small protein here is the troponin. So troponin will be released into the blood when there is a death of the cardiomyocyte. In this case, the increased blood level of troponin is the golden standard to diagnose a heart attack. So In other words, to say so, if a person, if a person walk into the ER claiming chest pain, how the first lab test they receive is check if there's increased troponin in the blood. So this slide is one of the most important thing in this topic. Let's fastly recap everything we talked about. The myosin and actin next to each other. The interaction between the myosin and actin can happen automatically. Whenever the myosin can directly bind to the actin, it automatically drags actin inward to make the sarcomere shorter. And so how do we regulate the muscle contraction? In most cases, when the muscle is relaxed, actin is blocked by tropomyosin. And when we need a muscle contraction, troponin is activated to remove tropomyosin from the actin so that the myosin can bind to the actin to drag the two group actin inward to make the whole sarcomere shorter. In that case, in the case of a heart attack, the golden standard to determine if there's any cell death of the heart muscle cells is to see whether there is elevated troponin in the blood. So next slide. So let's move on. Let me know if there's anything unclear here. Next thing, then we talk about how the troponin is activated. The troponin activation is based on the elevated calcium concentration in the cytoplasm. And in this case, in the heart muscle, cytoplasm is not called cytoplasm sometimes. They call it a sarcoplasm, sarco something is normally refers to something about the muscle cells. And then, so what happened is that in the case of troponin, there's three subunits in the troponin, and one of the subunits troponin particularly bind to calcium, and the calcium bind to the troponin, change the troponin shape, and then make the troponin remove the tropomyosin from the actin activation site. So now we know that it's the elevated calcium concentration in the cytoplasm that activate the troponin. And the next question is, what factors contribute to the elevated calcium in the cytoplasm? And then there's another key issue here. This is an internal circulation. And what has happened, the cells does not really acquire calcium from outside. It actually store calcium inside in this structure called sarcoplasmic reticulum. And in the last semester, probably you still remember this endoplasmic reticulum. That's ER. In the regular cell, it's called ER. But in muscle cells, we give another name, sarcoplasmic reticulum, or SR. So this is the pool. This is the storage of calcium or other things, mainly calcium here. This is the storage of calcium inside the cells. So in this case, the heart does not largely rely on the extracellular calcium because it needs a lot of calcium to release into the cytoplasm. So that rapidly increase the calcium ion concentration in the cytoplasm will activate the troponin, and the troponin will remove the tropomyosin so that the myosin can grab actin. All right, so now our story keep going upward. So now we know it is a calcium ion that activates the troponin. So and then what kind of factors that tell the SR to release the calcium. This is the cap of the calcium. This protein called RYR2. And this is a very important protein. It would be great if you can remember the name, but it's okay if you don't, because I don't think I have any question I would test it in my exams. But basically, what's the function of RYR? Imagine so it is a SR is the pool or a bottle. You contain a huge bottle of calcium inside. And how you can release the calcium from your bottle? Take off the cap. And RYR is the cap. So RYR open to release the calcium into the cytoplasm. Now we have the next question. What signal trigger the opening of RYR2? This signal is calcium again. So RYR is very close to the surface of the cell membrane. And then in the cell membrane, there's another channel that can release the actual cellular calcium very fastly into the cell. And in this calcium, if you only open this channel, this calcium channel on the surface of cells, this calcium itself does not reach the high level, high concentration enough to activate troponin. However, it says these two proteins are physically very close to each other. So literally when this channel open, this calcium literally blow into the RYR and forces to open. So this in this case is what triggered the RYR2 to open? That is the cell membrane calcium tunnel. Now we have another question. What opens? What opens the cell membrane calcium channel? We actually will stop here. What opened the cell membrane calcium channel? That is the action potential. I'm not sure you still remember what is the action potential, or you can say the depolarization. We'll talk about this concept in the next topic. And as of now, if you can remember that this is the depolarization, that'd be good enough, or action potential, that's good enough. So let's recap the whole thing. We basically talk about this whole procedure from the last event to the first event. But now let's talk about this in the forward the order now. Let's quickly recap this two pages. Say what happened first. This action potential reaching this cardiomyocyte. And then the action potential open up the membrane, the cell membrane calcium channel, and release a lot of calcium ions directly into this protein called RYR on the surface of the SR, the sarcoplasmic reticulum inside the muscle cell. And then once this RYR is open, it releases way more calcium ions from the SR storage all the way into the cytoplasm. And then the increased level of calcium ions in the cytoplasm, they will quickly bind to the protein called troponin. The troponin is a protein that holds the tropomyosin to block the myosin binding site on the actin. So when the troponin is activated, it removes the tropomyosin from the actin so that the myosin will bind to the actin. When the myosin bind to the actin, these two proteins really love each other. The myosin just grab the actin inward to the center of the sarcomere. And then because it has two arms, both arms bring the two actin fibers closer to the center. The whole sarcomere becomes shorter. And then not only one sarcomere, every single sarcomere in this cardiomyocyte becomes shorter, and this single cardiomyocyte becomes shorter. Not only one cardiomyocyte, in the ventricle or in the atrium, all the cardiomyocytes, they contract at the same time. So every single cardiomyocyte contract, becomes shorter at the same time, and then either the atrium or the ventricle, the whole thing contract, squeeze, becomes smaller, squeeze blood outside the ventricles. This is how it happens. Any question? Okay, let's move on. And next thing is, we already talked about this in the lab. Just imagine why our heart never feel tired. There are many reasons for this. One of the things is actually here. This RVR2 is between the interaction between RVR2 and the calcium, there's no directly physical. So RVR2 actually lasts much longer than RVR1 in the skeletal muscle. And another thing is that the cardiomyocyte has a much higher efficient use of energy than any other cells in our body. It constantly uses the fatty acids. So the primary fuel for the cardiomyocytes is fatty acids, and it utilizes a larger proportion of aerobic. I hope you still remember what is aerobic respiration. Aerobic respiration is that you utilize the oxygen and to acquire the maximum level of ATPs from glucose. And at the same time, if you want to obtain ATPs from fat acids, you have to use the aerobic respiration. And where inside the cells, where does the aerobic respiration happen? Anyone where does the oxidation happen in the cells? What does organelles mediate the oxidation? Anyone? Yeah.
What's the question?
Mitochondria. Which? Yes, mitochondria. I was asking which organelles in the cells, which organelle in the cells is the place for the oxidation or aerobic respiration to happen? The mitochondria. And in these cases, which cell has the highest content of mitochondria? The cardiomyocytes. Cardiomyocytes has a very large number. This one, mitochondria. Mitochondria is the cell has the highest number of the mitochondria, and muscle cells has the second one. And so there's one thing, key word, key knowledge here is that the cardiomyocytes use the more than half of the energy that you use by the cardiomyocytes comes from the fatty acid. And the next question is, what does which step does the cardiomyocytes need energy? The one thing here, let's coming back all the way here. This step from this the myosin drag the action forward. This is the final step that making the sarcomere or the cardiomyocyte shorter. This step, you can say it consumes energy, but if you just talk about ATP, this step does not really release ATP. It's more like, with a bowl, right? A bowl to release arrow. So you draw the bowl. Now you have the energy, give the energy to the bowl and release it. The released point, actually, you do not need any force to give it. You just let the bowl to return its own shape to drive the arrow forward. So in this case, this is the last step. You basically release the bone straight to let the arrow go out, to release the arrow. And so this step actually does not really require energy. And what really releases the energy? That's because after your contraction, you have to reshape the myosin into, we call it the poised status. So you have to redraw the ball. So the same thing, just imagine the ball. You release the arrow, and then you have to redraw the ball again. And this redraw the ball, that's the status to make the myosin It's mouse and drag forward, and then you need ATP to put it back to the poised status. That's one of the things that you need energy you need ATP in the heart to do. So to re-poised the mouse to return the mouse in the high energy status. That's one thing. Another thing is here. When the artery are open, it releases a large number of calcium into the cytoplasm. And for a better for this the heart cardiomyocytes to contract in a rhythm, you need to remain after the one contraction. You need to reduce very fast, very rapidly to reduce the calcium ion concentration in the cytoplasm. And how you do so? There's a pump here. The pump actively pumping the calcium back into the SR storage. So this is another major way that you need ATP to make it work. So the second way. And then another thing we talk about here would activate the calcium channel on the surface of the cell membrane, the action potential. And the action potential happens through depolarization. And then this action potential is another way like drawing the arrow. Next, you have to gradually reestablish this resting potential. We'll talk about the resting potential in the next topic. But here, if you do not remember, just keep try your best to memorize this. We'll talk about in the next topic. The third way to use ATP in the cardiomyocytes is to reestablish the resting potential. And then, so let's briefly review this thing here. There are mainly three ways that the cardiomyocytes need ATP to reposition the myosin in the poised shape, in the high energy status, to reabsorb the calcium ion back to the SR from the cytoplasm to the SR, and to reestablish the resting potential. It's all here. It's all here. To reestablish the resting potential in the cell membrane. And okay, almost done. Then, since we talk about the cardiomyocytes, the heart is an organ. So heart does not really, they last really long. There's the one of the good feature of the heart muscle. But another side of the cardiomyocytes, they rarely regenerate. So in general, in a how to call it, regular adult, every year there is about 1 to 2% turnover of cardiomyocytes, and also there's no stem cells in the heart. There's no stem cell in the heart. The turnover, the regeneration of the heart, mainly coming from some of those cardiomyocytes. They can gradually replicate. It's amazing. They can replicate in a very, very low status. So every year there's only 1 to 2% regeneration. And here I'm showing some of those research. If say exercise actually can increase the regeneration rate of the heart, and also prevent this regeneration rate actually gradually reduce along with aging, and then exercise can also prevent the loss of regeneration rate along with aging, even though it's very low, but it's still very essential to maintain a healthy heart. So because it's very low, in some cases, like we talked about earlier, in a heart attack, some region of the heart lack the blood supply, and then if the blood supply lasts longer, some of those cardiomyocytes will die. And what happened? Okay, here. Yeah. I thought it's in the next topic, but never mind. All right. So in this cases, let's back to the here. In a heart attack, in a heart attack, um, firstly, whenever people feel the chest pain, it's likely there's the hypoxia, the lack of blood supply in some region of the heart, but it does not really always lead to a heart attack. If this hypoxia is solved in time, no cell die and it's not a heart attack. However, if some cells start, if this hypoxia period lasts longer and some cells died, because the low regeneration rate of the heart, these dead cells cannot be replenished in time. And then you leave kind of a hole in the heart. And because you do not have you cannot provide the cardiomyocyte there in time. What happened here is that you cannot leave the hole there. Then another cell coming, they call fibroblasts. And fibroblasts coming here to deposit a lot of collagens. And sometimes at the same time, macrophages also coming here to remove the blood clotting and other stuff. In these cases, what happened here is that the macrophage may further activate the fibroblasts into a very active status. And then the fibroblasts just deposit tons of collagens into this site. And then this collagen becomes so thick and they cannot be penetrated further by other stuff. And this thick collagen stay there forever. And this is commonly we call it a scar tissue. And the scar tissue, as we mentioned in the last topic, cardiac output, the scar tissue cannot contract. It permanently reduces the elasticity and the contractility of the heart. And that's why, in most cases, a heart attack will leave a permanent scar tissue in the heart and also a permanently reduced heart function. So this is let me see what's this. Yeah, this is some research. I mean, I don't think I have anything in the test. In the research area, some people trying to fix this issue, and how you really can using a medical approach to stimulate heart regeneration. There's some potential research. One of those here is that to reactivate the heart stem cells. When we were young, we do have heart stem cells, and we lose them. And some people believe that in the adult heart, there's still stem cells there, only that they are quiescent, sleeping. So they are trying to give some stimulation to the heart to reactivate stem cells, so it can regenerate gradually. These stem cells, at least they hope, can gradually remove the scar tissue to restore at least a partial heart function. And in some tissue, actually, the scar tissue can be removed. It's not like whenever you generally have a scar tissue, it stays there forever. For example, and I often have a lot of scars on my hand. Some of those, given time, they disappear. That's because in our epidermis, that stem cells is super capable of regeneration. And then there's another approach direction of the research is that some people can find we call it pluripotent stem cells. These kind of stem cells can have the potential of differentiate into many other different type of cells, and these cells has the potential to difference into the heart muscle cells. In these cases, that's a dream of many of the researchers. They can get these stem cells from the embryo, and then they develop differentiate them into the heart, into heart cardiomyocytes. And when we have enough cardiomyocytes that derive from stem cells, we can make a patch onto this, and we cut off the scar of the heart and replace it by this patch of the cardiomyocytes, and we can almost perfectly restore the heart function. That's only a dream. Okay, so far it's not working yet, but probably in the future, 2 years, 5 years, or 20 years, it may happen. And here I'm showing this. These are the cells that they derived from stem cells. You can see this. They can actually contract by itself. You can contract by itself. And the problem is here, the darker color means, the red color means a stronger contraction. Currently, none of those cells that they acquire from stem cells is as strong as native cardiomyocytes. The upper level is the native cardiomyocytes. The lower panel is the stem cell derived. It's not strong enough. There's still many things to work on. And so that's it.